viral infection. So I did a little bit of a crude concept map up here, folks, the consequences of viral infection. So if I get infected today with a new virus, right? One possibility is that I'll have an effective immune response and my immune system will be able to eventually eliminate the virus. So I would think of myself as cured. I'm no longer carrying the virus, right? But in contrast, you guys, sometimes our immune system cannot eliminate the virus and we will remain infected for long periods of time. And a description of these infections that last a long time are chronic infections. And folks, what I did was I divided chronic infections up into two categories. And one is persistent. And by persistent, I mean as long as I'm infected, the virus is continuing to replicate. So the virus is always replicating, right? And a really good example of persistent chronic infections, you guys, would be hepatitis B virus that infects our liver, our hepatocytes, and it's constantly replicating. And this means that we constantly have really high concentrations of virus in our blood, right? So always really high levels of virus in our blood with hepatitis B. But you guys, another kind of chronic infection is what we'll call a latent infection. And in a latent infection, often what happens is that when we are first infected by the virus and it's replicating, we'll have a bout of clinical signs and symptoms, right? And then the immune system kicks in, and hopefully within a couple of weeks we'll start feeling better, and finally we might feel perfectly healthy. And we think, whew, thank heavens, I'm cured. I no longer have that virus. But what we're unaware of is the virus has not been eliminated. The virus is resting in some of our cells. It's not actively replicating, right? So we don't continue to have clinical signs and symptoms, but we're still infected, right? And you can think of, the virus is in kind of a dormant stage, right? It's not replicating.
Is that like a paralytic?
Some of them can be, so we'll come back to that, okay? Right, okay. So you guys, in these latent infections, what might happen is that later, there might be a couple of years later, or more than a few years later, there'll be some trigger that reactivates the virus, and it will start replicating again. And as a consequence, we'll have a whole new round of clinical signs and symptoms. So this isn't a new infection, it's a reactivation of that original infection, right? And you guys will see that there's a number of viruses that, this is a partial list, folks. So three examples of viruses that can cause these latent infections. So the herpes virus family, you guys, they're notorious for causing latent infections. And two examples that we'll discuss, you guys, are the chickenpox shingles herpes viruses, also called varicella zoster for chickenpox shingles. And then another group of herpes viruses, you guys, the human herpes viruses, type 1 and type 2. Historically, they were called the herpes simplex viruses. These little darlings, when they infect us, right, they have initial clinical signs and symptoms. Then they invade our sensory neurons, and they just lie there dormant until some trigger comes along. And it might be suppression of our immune system will trigger them to start replicating again, and we'll have a brand new round of clinical signs and symptoms. The human papillomavirus, you guys, a so-called wart virus, is another virus that can cause latent infections. We think of wart viruses, you guys, or warts as these solid collections of cells, right? It might be that we develop a wart, right? And then our immune system can kick in and we'll see the wart regress, but what we're not aware of is that the cells at the base are still infected, so we have like latent infections. So you can have a wart that will reappear at the same site.
Yeah. So our immune system doesn't respond to the infection unless the virus is replicating?
Well, we will see that the cells of the immune system have to be have to be triggered. They have to become aware that we're being invaded by a virus. And usually when we're first invaded by a virus, there will be an immune response. But viruses can hide out inside cells, and especially cells of the nervous system. Very often it's hard for cells of the cells of the immune system to detect pathogens that are hiding within our neurons. It's often kind of like an immunologically protected area for that. Yeah. So we'll give some examples. And then, folks, we'll see next week when we talk about HIV, that HIV can cause latent infections. You guys will recall we said the reverse transcriptase takes the HIV RNA and makes a double-stranded DNA copy of the HIV, which we call the HIV provirus. And we said that provirus gets inserted into our chromosome. Well, there's two fates for that cell that's infected with the HIV provirus. It might be that the cell will not start replicating HIV, right? Or the cell can start replicating HIV. The bad news is even in the cells that aren't actively involved in replicating HIV, they are still permanently infected, right? So we could say those cells that don't start replicating HIV right off the bat, they're latently infected, right? Okay. So let's see, you guys, let's see if we can see a couple of examples here. So let me go forward one slide, you guys, just so I can, okay. So folks, with latent human or animal viruses, there's different strategies. I think what Lindsay was asking, in some latent infections, the viruses actually get their genetic information into our chromosomes. But that's not always the case with latent viruses. It might be that they're just remaining, again, in kind of a dormant stage in the infected cell. And what we're going to do, you guys, is use the human herpes viruses type 1 and type 2 to kind of introduce this concept. And these human herpes viruses type 1 and type 2, you guys, are probably relatively widespread. And historically, you guys, I always laugh at this because it's like, you know, we write the textbooks and the viruses don't read. So, but anyway, historically, you guys, human herpes virus type 1 was described as causing infections above the belt, meaning mouth, fingers, you know, eyes, skin of the face. And human herpes virus type 2 was usually responsible for infections below the belt, usually meaning sexually transmitted infections. But you guys, it's so important for us to remind ourselves, do viruses read textbooks? No, they don't. And so it's really important to remember, you guys, that type 2, human herpes virus type 2 can cause oral, ocular infections. And type 1 viruses can cause infections of the genital tract, right? Because the viruses really don't care what we say they should do. The first infection, you guys, is when we first become infected, and the viruses will replicate within our cells. They usually cause lytic infections, and we'll end up with these fluid-filled vesicles, and the fluid is chock full of the infectious virus. And again, you guys, it might be maybe 2 to 3 weeks before, you know, the immune system responds, and these vesicles will break open, they'll crust over, and eventually heal. And you're like, whew, thank heavens, right? These are very painful infections. But what we're not aware of, you guys, is that these herpes simplex viruses, what they have done is they've invaded the axons of our sensory neurons. So they travel up the axon, the sensory neuron, and they lie dormant in the sensory neuron cell body, the portion where the nucleus is located. And they can remain there dormant for months, for years, until there's some kind of trigger. And very often, you guys, it's a trigger that lowers our immune defenses, like long periods of stress, which I know most of you experience. Chronic stress, that suppresses the immune system, right? If it was maybe a cancer patient undergoing radiation or chemotherapy, right, is gonna suppress the immune system, and that can cause reactivation. A transplant recipient, right, they're under immunosuppressive drugs, so those could all be triggers. So when the virus receives the trigger, right, it reactivates, and what that means is it's going to start replicating again. with a human herpes virus type 1 and type 2, what they do is they travel back down the sensory axon and they invade the epithelial cells that the sensory neuron innervates, right? And again, we get this lytic replication, so you have a whole new group of clinical signs and symptoms. And again, folks, that reactivation, it's not a second infection, it's a reactivation of that primary infection, right? So with herpes, simplex viruses, you guys, that infect the mouth or the eye here, or skin of the face, the viruses hang out in the trigeminal ganglion. So like right now, you guys, in college, I had oral herpes of the mouth. So ever since then, I will be lifelong carrying the herpes simplex virus up in my trigeminal ganglion. And I know for myself, you guys, usually at least once a year. Usually it's like maybe at the start of the semester. Very often it's right before I do the virus lectures. My virus reactivates. It starts to, it almost feels like it's marching down my sensory neuron. It starts to tingle a little bit. I go, oh no, here we go again. So usually I have a nice herpes reactivation lesions for you all to look at during this talk. So for some reason it didn't happen. It happened about a month ago instead. Okay? And during that reactivation, you guys, again, we are infectious. We're shedding virus. And very important, you guys, they've done experiments where they've discovered that we actually start shedding virus before we experience the clinical signs and symptoms. So we're probably shedding before we're aware that we're having reactivation. Let me see here. If folks, we get infected in our fingers, and this is an occupational hazard for those of you going into any aspect of dentistry. Like for me, you guys, let's say I go to the dentist to have my teeth cleaned, and I'm shedding herpes viruses, and I don't know it, and the hygienist doesn't know it. And let's say he or she's cleaning my teeth and using really sharp instruments, right? And they're gloved up, right? But those sharp instruments can pierce. So if I'm shedding herpes, it would be in my saliva. So if the hygienist, pokes through the glove and into the skin, and my saliva with the herpes virus gets in there, they can get their fingers infected. And that's called herpetic whitlow. And again, you guys, it's occupational hazard of folks in the dental field. And what will happen is after replicating in the epithelium, the same story, you guys, over and over again. The herpes virus invade the sensory neurons, and they'll hang out in the brachial ganglia, these collection of neuron cell bodies. And then again, when trigger reactivates the virus, they travel back down, and then again infect the epithelial cells, replicate, causing very painful lesions. And the same story is true, you guys, in when we have sexually transmitted genital tract infections with herpes. Same story. Initially we'll have clinical signs and symptoms, the viruses invade the sensory neurons, and then they lie dormant in the sacral ganglia. And again, they can be reactivated. Very importantly, folks, again, just to emphasize, they've done studies with folks that are latently infected with genital herpes, and they've shown that, that folks that have the latent genital tract infections with herpes can be shedding the virus without being aware. And it's just important for us to know, you guys, so that we're careful protecting the partner with whom we are intimate, right? Because we don't, want to try to avoid the herpes virus being spread to those with whom we're intimate. These are just some examples, folks, of consequences of some of the human herpes virus type 1 and type 2 infections. So some common names, you guys, for these reactivation of the herpes virus sometimes are called fever blisters or cold sores, often associated with getting sick, right? This is really frightening, you guys, because this is ophthalmic herpes, where the herpes invades the eye. And again, you guys, with repeated reactivation, ocular herpes can actually lead to blindness, so this is something we want to be really careful of. This young man, you guys, is a wrestler and has what's called herpes gladiatorum. So after the Roman gladiators, the wrestlers, right? So this is an occupational hazard if you're involved in sports where you have close physical contact with an opponent, right? So if your opponent is shedding herpes virus, right? And let's say you're wrestling, right? You get abrasions to the skin, and so you can get inoculated with the herpes. And this to me, you guys, is so disturbing because the infection is so widespread. And what to me is really, really worrisome for you guys is the eye is infected. So right now, he's gonna be in danger, kind of lifelong, of reactivation of that ocular infection, right? So again, we really worry about the potential for that to cause blindness. You can see it's spread down to the mouth, and it says here in the description that he had it on the neck and the shoulders, right? So he was really kind of massively inoculated. Here's the herpetic whitlow, folks. The vesicle caused by inoculation of the fingers, and again, this is the occupational hazard for anybody involved in dentistry. And here, folks, we see these really painful lesions in the genital tract, and again, these are fluid-filled vesicles, you guys, chock full of infectious virus. Yeah. And very painful, you guys. I mean, when my herpes reactivates, it is so painful, very painful lesions. Of concern, folks, is that if we have a pregnant mom and she has a genital infection with the herpes virus, there is concern because if she delivers her baby vaginally, there's great concern. What if the baby becomes infected, right? What if mom is shedding at the time of the vaginal delivery? And we know, babies' immune systems aren't fully developed when they are born. So the worry would be, what if the newborn baby becomes infected with the herpes virus and it just spreads everywhere? So in some babies, you guys, it'll cause skin infections, cutaneous ocular infections, infections of the nervous system, and in some instances can actually kill the baby. This range, you guys, of mortality rates, I was like, wow, that's a huge range. So this percent mortality, you guys, means for every 100 babies who get infected, how many die? And so this is a huge range, you guys, 30 to 80 per 100 babies that get infected with die. And I was like, why this big range? And one thought, you guys, might be, it might have to do with if mom has been infected for quite a while. Because if mom has been infected for a while with the herpes virus, this has given her immune system a chance to start making antibodies against the herpes virus. And you guys, this wonderful class of antibodies that humans make is called IgG. And mom can pass her IgG across the placenta to the baby. So if mom has circulating antibodies against herpes virus, she can transfer those to her baby. So when the baby is born, you guys, the baby will have mom's maternal antibodies circulating, and we would hope some of those antibodies might be neutralizing antibodies. So possibly in those babies, there's a lower chance of them becoming seriously infected, a lower chance of them dying. My concern, you guys, would be in a mom who just very recently has become infected with the genital herpes, because it might be she hasn't had a chance to produce those protective antibodies. There's not been a chance to pass them to the baby. So when the baby is born, truly the baby has no defense against these viruses. When I first started teaching, I would say, well, so if a pregnant mom has genital herpes, right off the bat, they're going to do a C-section, a cesarean delivery. But this is what's so good about teaching, you guys. I learned, I think, more from you than vice versa. So one of your colleagues worked in an OBGYN office, and she goes, that's not true. She goes, you can work, the parents can work with the physician and monitor for shedding. And she says it is possible, to have a normal vaginal delivery if mom is infected with herpes virus. You just want to be monitoring to check for shedding. But then another colleague in the class said that the hospital where she worked, it was almost like a hospital policy that if a pregnant mom had genital herpes, they were just automatically going to do a cesarean section. So I think the point is, you guys, that this is something, if you find yourselves in this situation, you want to talk with your healthcare provider, find out what your options are. And then another colleague many years ago, you guys, she shared with me that she had genital herpes, and the physician had put her on acyclovir, which won't cure you of herpes, but it really reduces replication rates. So you're not going to be replicating so many viruses, right? So you're not going to be shedding so many viruses. And another colleague looked into it and shared with me that acyclovir can be given to pregnant women. So that might be another way to protect a newborn if mom has the genital herpes. If acyclovir is safe to take, that's going to decrease the herpes virus replication and really decrease the chance of the baby becoming infected. Right? So just good to know, you guys, that there's different options. Oh, and I know one thing, you guys. I almost always forget to share this. So another way a newborn baby could become infected is like a person like me. You guys remember, I'm latently infected. So once or twice a year, my herpes virus reactivates, and I'm shedding the virus in what? My saliva, right? So if there was a newborn baby, my instinct is to do what? I just want to kiss it and just go, oh, the most wonderful thing in the world. But you guys, if I'm shedding virus, right? I mean, I might be the source of infection of a newborn. So just try to keep that in mind, you guys. I've always thought that could be really tricky. You know, like let's say it's a grandparent or an aunt or an uncle that wants to kiss the newborn baby. It's like that could get a little dicey. It's like, I see you might have herpes there, so I don't understand. Like that would not go over well. So I thought, you guys, maybe this is so silly, but I thought maybe like, you know, how nowadays when we have an outbreak of influenza, we say don't shake hands, you don't bump elbows. I thought, well, maybe we should start a tradition that we don't kiss the baby's head, we kiss toes instead. Yeah, those toes are so cute, you know. Anyway, yeah.
I was reading in the text for review that you had that 60 to 90% of adults have a latent herpes.
Yeah, it is amazing, you guys, when you start reading about the prevalence of a lot of these.
That's so many people.
It's really, really high. Well, and also, you guys, I mean, to me, and this is probably giving you way too much information, but if I think about it, like when we were growing up, your parents probably didn't do this, but when my brother and I were growing up, if we were out in public and we got dirty, you know, like our face was dirty, my mom wanted us to look presentable, so she'd give us a spit bath. Right? You know, she'd pull out her handkerchief and spit on it and clean us up a little bit like that. And I thought, that's probably another great way, you know, to spread herpes. So, I just think with herpes virus being present in saliva, and I mean the people we love, our kids, we're kissing them all the time, right? It's kind of, to me, maybe amazing that there's people that haven't become infected with herpes, right? So, and the other thing to remember, folks, is that the immune system can keep these viruses under control, right? So some people maybe get infected and don't have really severe clinical signs and symptoms and are latently infected and never know it and never have any reactivation. So a lot of us might have silent infections, right? It's just, I think the big concern is, especially when the immune system is severely depressed, you know, like cancer, transplant recipients, folks on immunosuppressive drugs, maybe long-term steroid for inflammatory disease. Those are folks that I should worry about. Yeah, you know, it is amazing. You know, we are microbial zoos. And folks, this was just, again, many people are so surprised to know that chickenpox and shingles are herpes viruses. They belong to the herpes family of viruses. Chickenpox is kind of misleading because it makes it sound like they're pox viruses. But you guys, the chickenpox, shingles, the varicella zoster virus, they share the same behavior as the human herpes virus type 1 and type 2. So initially, you guys, back in the day before the vaccination, chickenpox was a pretty common childhood disease. And in chickenpox, you guys, this would be the primary infection with the varicella zoster virus. It's usually by the respiratory route. You know, so there's respiratory infection, and then there's viremia, the virus is in the blood, and then the actual vesicles that we see on the skin, that's kind of like a later stage. So in chickenpox, you're infectious not only through your respiratory secretions, right, but through the virus in these fluid-filled vesicles, right? the same story as with, human herpes virus type 1 and type 2. You know, eventually the immune system responds, the vesicles break open, they crust, they dry, they fall off, and then you feel like, oh, thank heavens, you know, no more of that. But the same scenario, guys, the varicella virus, it invades the sensory neurons and hangs out in those ganglia, so that later on, right, if there's some kind of trigger, often, again, it's chronic stress, or classically, you guys, it's old age. Like, you know, in theory, I think they say at age 55, your immune system starts going downhill. Those of us that got infected as kids, we can reactivate, and when it reactivates, folks, we refer to it as shingles. And classically, it's described as causing these band of painful, fluid-filled vesicles, maybe across the chest or the shoulders, but that's so misleading, you guys. It can occur anywhere. It can occur on your arm. One of our students was fascinating, guys. We gave this talk, and she came up after the talk, and she showed me her finger, and I was like, have you had chicken pox before? She goes, yeah, I had it as a kid, and it looked like she had shingles on her finger. It looked like herpetic whitlow, but I think it was shingles, and she had it on her finger. It was fascinating. So you guys, with shingles, this came up yesterday, like, you have infectious viruses in these fluid-filled vesicles, but unlike chickenpox, you aren't infectious by the respiratory route. So it would be direct contact, somebody coming into direct contact with the fluid in these vesicles, and then maybe doing this or that, you know, inoculating themselves. That's how they would become infected. And this came up yesterday, you guys. You're saying, well, what if grandma or grandpa has shingles, and you have a child who's not been vaccinated against chickenpox, and who has not had chickenpox? You know, again, just we want to be careful that the child doesn't come into direct contact with those lesions, because in theory that might cause an infection. Probably, I kind of doubt, you guys, it happens very often, but just to be aware, right? Yeah.
In that example you just gave, if the child was infected, would they exhibit symptoms of shingles or chickenpox?
The first infection, the first, yeah.
Even
sorry,
if they
the initial
become infected
infection,
from somebody
yeah,
with shingles.
it's going to present as chickenpox. So by definition, shingles is the reactivation of that varicella zoster latent infection. Does that make sense, you guys? Okay. Yeah.
Do they still do those chickenpox clinics? Remember, like they would, like if one kid would be infected with chickenpox, and the parents would bring all their kids.
No, it's true, you guys. It's so true. We have chickenpox parties growing up, right? And actually, you guys, you're like, why would you do that? And this is actually a theme that we see with a lot of childhood diseases, and so one hypothesis is, you guys, with a lot of these infectious diseases, you want to relatively mild infectious diseases, you want to get them as a child, because often a lot of the damage that's done is done by your immune response. So one hypothesis is, if you get the infection as a child, you'll have a protective immune response, but you won't have an inappropriate immune response. Whereas with some of these so-called childhood diseases, if we get them when we're older and we have this strong, robust immune response, when you're older you get infected, you have such a strong immune response that that's going to actually cause damage, like the immune response causes damage. So I think that was the idea behind having these chickenpox parties, you guys, when we were little. The idea was parents wanted their kids to get infected early, and then you're protected. Well, you're carrying the virus, but anyway, you're protected, you know, wouldn't have a really severe immune response later on. The problem is, you guys, with that whole idea was that, that sometimes kids get really sick. And in fact, growing up, I remember in second grade, my best friend, she got chickenpox, and she was so sick. I don't know if they had to hospitalize her, but she was really sick, like to the point where a parent, I would be really worried. And so, like that's why, actually, guys, I would be, it's like, I think, having experienced that, seeing a child, you know, knowing somebody that got so sick with chickenpox, I'm not sure if I'd be keen on purposely exposing my kid. I mean, now there's the option of the vaccine, you guys. So how many of you have been vaccinated against chickenpox? Okay, right, okay, right, yeah. So what you might wanna do, you guys, that have been vaccinated against chickenpox, is just touch base with your healthcare provider to see, is it necessary to get like a booster vaccine, right? Just, have any of you asked that, or have your healthcare providers talked with you about the need to get a booster chickenpox vaccine?
After a couple of years, you have to get a booster for it.
You, okay,
After a
interesting.
certain amount of years.
Very, thank you, yeah.
I actually had chickenpox as a kid.
Yes.
So would I need to get a booster?
Well, luckily now, for those of us that had chickenpox, they have a shingles vaccine. So you guys, the shingles vaccine, it won't cure us, right, of the varicella zoster virus, but what the hope is that it will shorten the duration of the painful lesions, lessen the severity of the, it's almost disabling in some people, it's so painful. So reduce virus replication, reduce virus spread, reduce pain, shorten the duration of the reactivation. So I got my shingles vaccine because I got infected as a kid. But you guys, just right here, this is a topic that we'll come back to in the immunology section, and I just want to throw it in here because I think when I received my shingles vaccine, they forgot to tell me it's a live attenuated vaccine. And this is, I think, really important that we're aware of the advantages and disadvantages of these so-called live attenuated viral vaccines. So when we talk about attenuation, you guys, we can think of it as like kind of like weakening or lowering the virulence of the virus that will be used as part of the vaccine. Now, it's live in that it's actively replicating. So when we're vaccinated, the vaccination mimics natural infection in that the virus is replicating. And the advantage, you guys, of these attenuated live virus vaccines, they tend to give you the best immunity and often the longest immune memory. Okay, so we get a strong immune response. And we get good immunological memory. So we have lots of good memory cells circulating. But you guys, the bad part about this, the dangerous part of this is that you should not vaccinate people that have underlying immune disorders, because if their immune system is suppressed or depressed, what do you think might happen? Yeah, the attenuated, low virulence vaccine viruses actually can cause disease in folks that are immunocompromised. You guys, the next slide, I found this slide. I'm gonna just give some examples, folks, of where you might want to avoid giving attenuated live viral vaccines. So folks, if I have HIV right now, you guys, I could still get an attenuated live virus vaccine. It's only when I progress to AIDS, right? We'll find out, you guys, that HIV destroys the most important cell of the immune system. Oh yeah, I'm so sorry. No. For the students. No, Carmen's helping design the new microbiology lab in the new building, so maybe your kids or grandkids will have a beautiful microbiology lab. Right? So you guys, so again, going to some situations where we would not want to use live attenuated viral vaccines. So again, you guys, if I'm HIV positive, then my immune system is still fine until the HIV destroys enough of my T helper lymphocytes, and we'll see, you guys, that's the most important cell in the immune system. And it's at that point, you guys, that I then officially have AIDS, the acquired immune deficiency syndrome. And so it's, if your patient is HIV positive and has developed AIDS, you wouldn't want to give the live virus vaccines because their immune system can't even handle that weakened vaccine viral strain. And again, guys, I won't go through the whole thing, but if we have cancers of the blood system, like leukemias or solid organ cancers, bone marrow transplants, it says contraindicated, so live attenuated vaccines during chemotherapy, don't vaccinate with live attenuated during chemotherapy. Here, what are they saying, you guys? Contraindicated vaccines. So here, measles, mumps, rubella. This is the varicella zoster. You don't want to vaccinate up to 2 years after the transplant, right? So just some special precautions, folks. Not only, you guys, do you want to avoid vaccinating people that are potentially, immunocompromised, what's really important for them to tell you is when you're vaccinated, remember the vaccine virus is replicating. So you are shedding vaccine virus for a while, right? And especially those of you going into medicine, you need to be careful because what if you're taking care of patients, right, that have these conditions and you're shedding the vaccine virus. So just remember that, you guys. And like I said, I think when I got my shingles vaccine, I don't think they told me, reminded me about that, be careful, who I'm around. Like, again, you guys, like a little baby, wouldn't want to be coming in contact with a little baby probably for, I'm thinking maybe a couple of weeks, right? Anyway, so always good, you guys, if you're getting vaccinated or you're getting your kids vaccinated, ask them, what is the vaccine, right? If it's live and attenuated, ask them if there's any special precautions you should take. Okay, all right. Onwards. So, this is kind of an introduction, you guys, into, I think this is the last topic in this introduction, folks. So, the broader topic here, you guys, is viral infections, which may increase the risk of cancer. And such viruses, you guys, that may increase our risk for cancer after we get infected are called oncogenic viruses. And I'm seeing here, looking at my slide, it's like, it is true that there is concern that viruses that can cause latent infections, some of them might increase the risk for certain types of cancer. But there's others, you guys, like hepatitis B virus, which is a persistent chronic infection, right? Hepatitis B virus can also increase the risk for liver cancer, right? So I think maybe instead of just latent, I should have said chronic infections. So it could be, latent or persistent with specific viruses, you guys. This isn't all viruses. So let me, and just on vocabulary, folks. So when cells no longer can control their ability to replicate, right? So if we have like human cells that get infected with these viruses, they interfere with the ability of the cells to control the cell cycle replication. They say that those cells are transformed. And you guys, I just want you to know that on the lecture exam, I won't use this term transform because it's the same term we use in horizontal gene transfer in bacteria. So this is in biology, because when you hear the cells have been transformed, you have to ask, wait, are we talking about cancer? Or are we talking about horizontal gene transfer in bacteria? So just to give you a heads up. And again, you guys, on lecture exam 3, I won't use this term transform because I don't want you to get it confused with horizontal gene transfer. But you guys, let's just look at a couple of examples of oncogenic viruses. So you guys, so persistent infections with hepatitis B virus and hepatitis C virus, because they're constantly replicating, our liver cells are constantly being destroyed. And you guys, our liver is so wonderful. It has incredible ability to, the hepatocytes have great ability to replicate, to replace damaged cells. But do you remember, folks, how we said all of our cells, they can only replicate so many times? And once they use up their telomeres, they can't replicate anymore, right? So in chronic infections, a problem might be that eventually the hepatocytes can no longer replicate to replace the destroyed cells. So then we end up with like fibrotic scar tissue, replacing functional liver cells. So we end up with liver cirrhosis, which can cause liver failure. And the only cure for that, you guys, is a liver transplant, which is just, oh my gosh, the risk of liver transplants and getting, a donor and all of that. And furthermore, you guys, if we think about it, every time the liver cells are ready to divide, they copy their chromosomes, right? And what do we know about DNA polymerase? It proofreads, right? But does it still have a mistake rate? It does. And so you could say, you know what, anytime our cells are having to divide more than normally, that by itself might increase the risk for mutations and thus cancer. So, and I know you guys know this already, but another serious consequence of chronic hepatitis B and C infections is the risk for liver cancer, right? So that's a tough one. You know what, you guys, let me just develop this a little bit, because I was really amazed at the differences with hepatitis B, you guys, the differences in the rates of chronic infections, healthy adults versus little babies. So let me just do this really quick, you guys. So if we're talking about hepatitis B infections, and hepatitis B, you guys, it is a DNA virus. So let's take 100 healthy adults become infected. The good news is, you guys, 90% of the adults have they develop an effective immune response. The virus is eliminated. And there's no increased risk of liver cirrhosis, nor of liver cancer. But you guys, in the other 10%, for, who knows, maybe it's the strain of the hepatitis B virus, maybe some strains are more virulent. 10% of the adults, maybe it has to do, you guys, with our genetics, right? With our host genetics. The immune system does not eliminate the virus. So we have these chronic persistent infections, and remember, you guys, with hepatitis B, usually it just keeps replicating and replicating, so we have really high concentrations of virus in the blood. Right, so wherever the blood goes, the virus goes. And then, so it's these guys, folks, that have the chronic infections that are going to have the increased risk of cirrhosis and liver cancer. But, you guys, the things are really different. If we're talking about a Hep B positive mama, a pregnant mom. So remember, you guys, this incredibly high levels of hepatitis B that's in human blood, right? So just imagine, you guys, a vaginal delivery and there's blood everywhere, right? So what happens if the baby becomes infected? And this would be what we call a vertical transmission, you guys, from one generation to the next, mom to baby. And we'll talk about this in medical micro, so this is called vertical transmission. And again, you guys, remember the baby's immune system when they're born their immune system really isn't fully functional. So here, you guys, if we look at 100 babies, neonates, we'll say neonates, newborns, the infection rates are the opposite, right? So only like 10% of babies will clear the virus, right? And what's happening to the other 90%, you guys? 90% of the babies, what happens to them? They become chronically infected, right? Yeah. And in some areas of the world, folks, the prevalence of hepatitis B is astronomical. You're like, what the heck's going on, right? 90% of babies become chronically infected. So in some parts of the world, you guys, the prevalence of hepatitis B is so high. And you're like, why is that? Well, one thought is, you guys, maybe many generations ago, right? Maybe that population got infected, and then we started this vertical transmission, mom to baby, right? So 9 out of 10 babies that get infected are going to develop chronic infections, and the girl babies are going to grow up and maybe get pregnant themselves, right? And pass that on to their babies. So, wow, this is such a bummer, you guys. But the good news is, folks, there's now a hepatitis B vaccine. And so now, you guys, with this hepatitis B vaccine, hopefully we can break this, this cycle of mom to baby, baby goes up, mom to baby. And I know that there is a lot of controversy, because I think, you guys, have any of you had newborns and they vaccinate their baby right off the bat?
Yeah.
Yeah, do they just boom, right off the bat they vaccinate? Because what they found, you guys, is that if you vaccinate the newborn, it does help prevent chronic infection. It does help prevent the chronic infection. And I know a lot of parents, and I totally understand this, you guys, a lot of parents are really concerned because the babies are getting so many vaccinations, and they're like, well, hepatitis B, what is the chance my baby's gonna get it? And my thought is, you guys, maybe they just figure it's economical, instead of testing all the pregnant moms to see if they're positive, so we're just gonna vaccinate all the babies. I'm not sure if it's like a cost benefit issue, but I think, you guys, this will be awesome, because in some parts of the world where prevalence is so high, vaccination might really help break that cycle of one generation to the next, to the next, to the next. Okay. HPV, you guys, so HPV, folks, are the so-called human wart viruses, and you guys, there's so many different strains. So the strains, you guys, that cause the solid lump of cells, usually those aren't the oncogenic strains. There's like just a few strains that are truly oncogenic, and the two strains, you guys, that I keep hearing over and over again are strain 16. Maybe this is the really most prevalent one here in the United States, and then strain 18. It's HPV type 18, you guys, was what was discovered in Henrietta Lacks's cervical cancer cells. Remember that? And the guy got the Nobel Prize for that, to show that HPV was the cause of the cancer in those cervical cells. So, you guys, these strains tend to be transmitted sexually, and the bad news is, you guys, is that when they infect like cervical cells, they increase their risk for cervical cancer. If they infect cells of the penis, they increase the risk for penile cancer. The same with anal rectal cells, you guys, increase the risk for anal rectal cancer. And another thing we're really worried about, folks, let me erase this, is they're starting to see a big increase in oral and throat cancers caused by these strains. And some epidemiologists, trying to figure out, why are we seeing all these new cases of oral and laryngeal and throat cancers with HPV 16 and 18? So we're going to call these oncogenic. And one hypothesis is that amongst young people, might think that oral sex is safer than regular sex, right? And certainly, you guys, you know, a woman can become pregnant through oral sex, but what people aren't aware of is the pathogens that can colonize the genital tract are very happy in our throat, right? So mucous membrane is a mucous membrane. And so the concern is that people don't understand that if they're practicing oral sex, you still have to have safe sex, right? Because you can still transfer these pathogens. So they think this is why we're seeing an increase in oral, laryngeal, throat cancers, right? Because people are thinking that the sexually transmitted pathogens will find a nice home, you know, in our throats, and they're just happy there, you guys. It's not good. We don't want them there. So, and again, you guys, we have an increase in cervical, penile is of the penis, anal, rectal cancers. But again, you guys, the good news is we now have at least 2 vaccines that can guard against infection with HPV 16 and 18. The first one, you guys, that came on the market was called the Gardasil vaccine. Yeah, remember that? Yeah. And you guys, Gardasil protects against 4 strains of HPV. So it includes the strain 16 and 18. And there's 2 other strains, you guys, and I forget the numbers, but they included 2 other strains of HPV that are common causes of genital infections. And then the second vaccine, you guys, I think it only has HPV 16 and 18. And folks, when it was initially licensed, it was only licensed for females, which to me was like, what, are you kidding? I mean, it's like, males are at high risk too. So luckily, finally, they got a license for males as well. And the problem is, I think sometimes people still think it's just for females. But you guys, it's so important that men, our sons, are vaccinated because, you know, throat cancer, cancer of the penis, anal rectal cancer, right? So, yeah. So I am really, to me, you guys, this is wonderful because we have a hepatitis B vaccine. We can consider this an anti-cancer vaccine. And to me, you guys, this HPV vaccines, these are like anti-cancer vaccines, you know, prevent the chance of, so just imagine if Henrietta Lacks had been, you know, she lived at a time when we had these vaccines, she wouldn't have died as a young woman, right? Okay. And then you guys, this one, on the exam, I usually don't ask about this one, but I just wanted to throw this in here, folks. This is another herpes virus that is associated with increased risk for cancers. And folks, the reason I did throw this in here is I think it's important for us to remember that in these viruses, there's different strains. Some are high virulence, some are low virulence. And the other thing, you guys, that's really important when we're talking about cancer is that we all have different genetics, right? And the reason I wanted to just give you that background is the herpes virus, the Epstein-Barr virus. This is the virus that causes infectious mononucleosis. I remember as a teenager, right? Oh, it's a kissing disease. Oh, so-and-so has the kissing disease, right? Because it's transmitted through saliva. But to me, you guys, it's so interesting because here in the United States, I don't think we commonly associate infectious mononucleosis with an increased risk for cancer. But in other parts of the world, the strains that are circulating, and again, you guys, it might have to do with host genetics, infections with Epstein-Barr virus can increase the risk for cancer called Burkitt's lymphoma, and also potentially life-threatening nasopharyngeal cancer. And again, you guys, I think it's because there's different strains of virus circulating, and again, the host that's being infected, our genetics, you know, our immune response is going to vary. So that one just really surprised me because again, growing up as a kid, you know, everybody got infectious mononucleosis, and I never thought that we would worry about it causing cancer later. Yeah.
What is the success rate of that vaccine?
I'm sorry.
Success rate of
Oh, okay, that's so good, you guys. And I I don't suppose anybody would wanna do an extra credit poster on, aren't I horrible, you guys? But yeah, you ask such great questions. I don't have those values. If you shoot me an email, I'll try to find them, right? Because I know that was a concern, you guys, when the vaccine first came out, and there was a lot of controversy. And again, you guys, I hope I'm not showing disrespect, but there was a lot of controversy when the Gardasil vaccine came out because parents were worried if they vaccinated their daughters, it was like telling their daughters it was okay to have sex, you know, to be promiscuous. And that, gosh, you guys, that's a whole big topic right there. And I know some people said, well, how do we know it will protect against cervical cancer? So the question you're asking is really a good one. You know, have they done the longitudinal studies to see, you know, the kids? I'm trying to think when our daughter got vaccinated. I think she got vaccinated in junior high. She's almost 30 now. So there should be many, many years of data to find out how many of the kids that got vaccinated did any of them go on to develop any HPV-associated cancers. So I don't mean to throw it back at you, and if nobody wants to do it, if you shoot me an email, because that's a good question, I should have it in, should be part of our slides, yeah. Okay, all right. And this one, you guys were just gonna, this one, I think this is a little bit of overkill. I mean, the whole idea of oncogenic viruses, it deserves probably several weeks of lectures. So I think you guys right here, let's see here. Yeah, and just wanted to end on a happy note, you guys, that I think our vaccines against the HPV 16, 18, we can say those are anti-cancer vaccines, so kind of a happy note. With that, you guys, we're gonna close our that was the introduction to viruses, right? And so, what we'll do next, you guys, is just get started on the second virus, PowerPoint. And we're just gonna look at a few RNA viruses, influenza virus, HIV, and a little bit on polioviruses. The reason, folks, I wanted to do a little bit more in detail, for example, on influenza virus. A few years ago, another microbiologist friend and I went down to the state public health reference lab in Vallejo, and it's a great, oh my God, it was so cool, you guys, just fascinating. And we asked the folks that work there, we go, what is the infectious disease that you're most worried about? You know, as public health microbiologists. And I thought they were gonna say something like Ebola, you know, something dramatic like that. And they said, what makes us worried and not able to sleep at night is influenza. So you guys, you know, this one might be the one that really comes in and has the potential to kill a lot of us. So I wanted us to have, you know, some background in influenza. So again, you guys are going to do RNA viruses, and we just want to remember, you guys, that we said there was like twice as many RNA virus families as DNA viruses, and what was our hypothesis to explain that diversity? Yeah, the high mutation rate, and why do they have higher mutation rates? RNA polymerase, which is going to copy their RNA, can't proofread, right? So we're going to get a lot more mistakes, many more mutations. We're going to have a lot more genetic diversity amongst our RNA viruses. Good. So you guys, the first two RNA viruses we'll talk about, luckily, thank heavens, they're envelope viruses. So influenza is envelope and HIV is envelope. Why do I say luckily? That's right. We said, you guys, that envelope has the adhesins, right? And remember, the envelope, you guys, it's like a layer of olive oil. It's very delicate. So when influenza virus or HIV is shed into the environment, anything that damages the envelope, it could be soap and water, it could be dry ink, right? Anything that damages the envelope makes the virus incapable of attaching to host cells. So voila, they're inactivated. So you guys, that's like, I think the only good thing I can think about influenza and HIV is thank heavens they're envelope, right? They're not going to remain infectious for long in the environment. And you guys, what we'll do is we'll start out with influenza virus. And there's three groups, you guys, of influenza viruses. into the influenza viruses, folks. There's 3 groups. A, B, and C. So the C viruses, you guys, they have such low virulence. We don't even include them in the vaccine. So low virulence, not in seasonal vaccine. We just don't worry about them. The group B's, you guys, these infect humans only, and they can cause serious illness. Sorry, infect humans only. Can cause serious infection. So usually in the seasonal vaccine, you guys, at least one group B is included in the seasonal influenza vaccine. The group you guys were really worried about are the group A's. So our whole lecture, you guys, is going to be focused on the group A's, because not only can these cause serious infections, one of the complicating factors is group A's can infect animals and humans, and many strains are what we call zoonotic. What does zoonotic mean? Yeah, many strains are zoonotic, meaning they can be passed from a non-human animal to humans. So have you guys heard of avian influenza? What does avian mean?
Chicken.
Yeah, literally means birds, right? So avian influenza, bird influenza. Do we know some strains can infect humans? Yeah, and can kill us. We're really worried about those. What about swine influenza? What does swine represent? Pigs, right? I adore pigs. They're such cool animals. But some strains of swine influenza, you guys, do you think they can infect humans? Yeah, they can. And we're gonna see, folks, because there are so many non-human animal reservoirs, this is further gonna increase the genetic diversity in the group A's, right? So, wow, genetically they are so diverse. Okay. Just a little background, you guys, the name influenza. Remember way back when we didn't have electron microscopes, nobody knew what viruses were. Remember how we said that there's a lot of horrible human epidemics, pandemics of infectious disease that were caused by virus, and because nobody knew what a virus was, you know, people would blame other people. You say, oh, it's your fault, right? Or some people said it was the influence of maybe misalignment of the planets, right? So influenza, influence of planet misalignment, right? Because nobody knew what was causing it, and that just added to the grief. But now, you guys, now that we have our electron microscopes, right, there have been a lot of cool studies done on group A influenzas. And so we'll just cartoon kind of superficially, you guys. So we know that they steal host cell membrane to make the viral envelope. Okay, and what's really interesting, you guys, is the group A influenza, it has 8 RNA segments. That carries all the genetic information of the influenza viruses. So you guys, I'll use blue, so 1, 2, 3, 4, 5, 6, 7, 8. So these are the 8 RNA segments that makes up the genome. And each segment, you guys, carries different genes. It's not like copies of the same RNA, they carry different genes. And folks, I won't cartoon it, but each RNA segment is then going to be covered in capsomeres. So like each one always has like their own little capsid there, but I just leave them naked. So folks, what do we know about envelope viruses? What do they have to do to the host cell membrane before they steal it? Do they have to insert viral proteins into the envelope? And what's one of the viral proteins they have to insert? a glycoprotein, right? And what would be the function? So the first step in infection, you guys, a virus has to do what to a host cell? Attach, right? So you guys, so what would be the function of one of the viral proteins on the envelope? Would it be the adhesive? Good, you guys. So what we're gonna do is we're gonna use little spiky things. So these are gonna be viral glycoproteins, a little sugar carbohydrate attached to proteins. So that's glycoprotein. And these little sticks, you guys, these are the heme agglutinins. And it literally means blood clumping. And the reason they were named blood clumping is it was noticed that when you mix influenza virus and red blood cells, the influenza virus binds to the surface of the red blood cells by their heme agglutinins, these little protein spikes. But that's not the function of this, you guys. The heme agglutinin is the influenza adhesin. So that's important, guys. We'll put a star by it because we want to remember that the heme agglutinin is the influenza adhesin, and it binds to host cell sialic acid. And this too, you guys, is going to be really important later when we talk about mortality rates, right? It's going to be related to where the sialic acid receptors are in a host. Okay. Now, you guys, the good news is the heme agglutinin, remember, it's a protein. And proteins are great antigens. And an antigen is a substance that will trigger an immune response. And usually the example we give you guys is antibody production. So kind of a great thing is that when we get infected with an influenza, our immune system recognizes that hemagglutinin is foreign and will make antibodies against them. So the hemagglutinin, you guys, is often called the hemagglutinin antigen. Hemagglutinin antigen. We can trigger production of neutralizing antibodies against it, antibodies that bind to the hemagglutinin and block attachment. So that's how our bodies naturally protect us, right? It's also one of the goals of the influenza vaccines. We're trying to trigger production of neutralizing antibodies against the hemagglutinin. So you guys, as biologists, we're really bad. Hemagglutinin, sometimes we just use HA, right? And folks, out there in nature, if we look at all the group A influenzas, there's at least 15, maybe 16 different hemagglutinins. 15 to 16 different hemagglutinins, and these can be detected by different antibodies. So I'll put HAs, okay? And we use the type of hemagglutinин to identify a specific strain of influenza virus, which we'll do here in just a bit, you guys. Now, in addition to hemagglutinin, influenza viruses will also in the envelope, they're gonna place another glycoprotein. And these, you guys, it's another protein. This protein is called neuraminidase. Okay, so it's also antigen. It sometimes is abbreviated as NA. And you guys, there's a couple of possible roles. For right now, you guys will just say, we'll try to keep it simple and say neuraminidase helps newly replicated influenza virus to escape from the host cell. Okay, so we'll say assist newly replicated viruses to escape from the host cell. And we'll have a photograph, you guys, that'll help us, hopefully. Okay, and folks, out in nature, there's at least 9, maybe 10 neuraminidases. So maybe about 9 to 10 neuraminidases out in nature. So, folks, we can use the specific types of hemagglutinins and neuraminases. Right? to identify specific strains of influenza A virus. So if you isolate an influenza A virus, you know, you're gonna be asked, what's the H and the N type? So you guys, this is an example of how these specific types are named. So we could have an influenza group A H1N1, or an influenza group A H2N3, okay? And we'll see that we associate different H and N combinations with different animal reservoirs. Okay? And then folks, again, just as part of this big overview, just as you guys shared with me, influenza viruses are RNA viruses, so they have potential for high mutation rates, right? So you guys, that means there's potential for little changes in the amino acid sequence of the hemagglutinin and the neuraminidase, right? Because of mistakes that RNA polymerase makes. These little changes in the amino acid sequence of the H and N antigens, you guys, is referred to as antigenic drift. And again, this is a result of the mistakes made by RNA polymerase when RNA polymerase is copying the influenza RNA. But you guys, something far more dramatic is a phenomenon called antigenic shift. Antigenic shift is when one host is co-infected with two different strains of influenza virus. And you guys, it's wild because a single host cell can be co-infected with two different influenza viruses. So you guys, we'll just make this up just really quick for right now. Let's say one host cell is co-infected, means infected at the same time, two different influenza strains. And we'll make this up, you guys. Let's pretend that you have your host cell gets infected with an H1N1 and an H2N3. Because the RNA genes that encode the H and the N antigens, you guys, I'll just put like HA gene here, and maybe the NA gene there. Because these genes are carried on different segments, when the host cell is copying the RNA segments of the two different viruses, right? And then remember, you guys, we have assembly, right? It's random which segments get packaged into the newly replicated viruses. So as a consequence, you guys, we can end up with 4 genotypes of newly replicated viruses. And can you help me out, you guys? What would be the 4 genotypes? We could do the easy ones first, you guys. Shall we do the parental genotypes first? Okay, so some of the viruses released would be the parentals, H1N1, and what else? H2N3, right? But you guys, we're gonna have some brand new assortments, right? Brand new combinations. Perfect, you guys, H1N3, I'll put it in blue, you guys, just H1N3, right? There's H1 and N3, and what would the other one be? H2N1, H2N1. So these guys are brand new, they're brand new combinations of H and N antigens. Major change, you guys, in the antigenic makeup of these recombinant viruses. So this big change, you guys, in H and N antigens is called antigenic shift. And it's the result of, we could say, recombination or reassortment between two different influenza viruses, right? Yeah.
Okay, so you were saying that if they do it like seasonally, right? This vaccine.
Uh-huh.
So how the heck do they figure that out?
Yeah, thank you, right? Yeah, they have to guess. And one thing you guys, okay, so remember we said that the inactivated influenza vaccine that's given annually, that one's grown in decades, and it takes 6 months to grow enough vaccine virus, right? So Lindsay's saying, how can they figure out which ones to include? And they have to guess. What they do is they look at the epidemic strains that were circulating last winter. They think, well, maybe we should include those. But then what they'll also do, you guys, is look at the southern hemisphere, because northern hemisphere and southern hemisphere, the seasons are reversed. Like right now, we're going into winter. They're going into summer. So what they'll do here in the northern hemisphere, they'll look at the strains that are circulating in the southern hemisphere. So like in spring, summer here, we're trying to figure out which ones. They'll look at the strains that are circulating in the southern hemisphere. They'll say, oh, this one's rapidly spreading. Maybe we should include it in our vaccine. So they have to guess, you guys. It's guessing, it's educated guessing. Sometimes they get it right, sometimes they get it wrong. So that's why some years, maybe you've experienced, or maybe a friend or family have experienced that they got the influenza vaccine and they still got an influenza, right? It might be that the vaccine didn't catch that particular strain of influenza. But you guys, this is me, and you guys, I'm a vaccine maniac. My thought is that even if they miss the exact strain, I still want to get vaccinated because I'm hoping there might be a little bit of cross protection going there, and especially an old person like me, we need all the protection we can get. So I run to the drugstore as soon as they say we have influenza. I run there and go, I'm paying out of pocket. I want my influenza vaccine right now. Yeah, yeah. So I'm still a big believer in it. Yeah, we just need a way, you guys, so that it'll be quicker, so it won't take 6 months. Right, so they'll have a better idea of what epidemic strains are circulating. That would be awesome. Okay, guys, so let's see what we got. This is my favorite slide. I'm just blown away, you guys, by the animal reservoirs of influenza virus. We'll end with this slide, you guys. It's believed that wild water birds like ducks were the original natural reservoir of influenza virus. And the reason is, you guys, when you isolate influenza viruses from wild ducks and geese, you can find every combination of H and N antigens. So it seems like they were the original hosts for the group A influenza viruses. And from them, the group A spread to all these other hosts. So they spread to domesticated poultry like chickens. And it's so fascinating, you guys. They found out that chickens that were infected with influenza virus, when the uncooked meat was fed to members of the cat family, the cats got infected. In zoos, tigers got infected, cats got infected. From the wild waterfowl, you guys, the influenza viruses can be spread to humans. And again, you guys, it blows me away. There's marine mammals, seals, and whales that have influenza viruses. This is a big one in horses. If you guys are horse owners, you probably vaccinated against influenza. Ferrets, you guys, this is a favorite lab model when they're studying human influenza viruses. I'm like, what? Ferrets and humans, that's an interesting combination. This one, you guys, on Tuesday, we'll come back and we'll talk about how pigs are such an important reservoir of influenza A viruses, because you guys, pigs can be co-infected with avian influenza, with human influenza, and with swine influenza. So they're what we call reassortment vessels. They're great mixing pots for brand new combinations of H and N replicated viruses. So you guys, so we'll start here. We'll finish influenza. We'll do HIV, a little bit of polio, and then if not Tuesday, you guys, Thursday we'll do prion. And you guys, if you